Hello, everybody. My name is Vittorio Venturi. I welcome you all to this CEDRIC webinar series that we have. I just have a few slides, just 3 or 4 slides to present our project. So the CEDRIC project is an Interreg project. These are projects funded by the European Union as well as the local regional governments. And the aim is to promote cross-border collaborations. And here we are located in Trieste, in the region of Friuli-Venezia Giulia. So we border Slovenia and Austria. And this is an Italian-Austria Interreg project. And the aim of this project is to really exploit soil microbiome biodiversity to promote agricultural development. The duration of here you see the duration of this Interreg is 3 years, between 2024 to 2026. We have a budget of 1.2 million euros approximately, and the EU financing is almost €1 million. We have 8 partners between Italy and Austria. The objectives are to, let's say, implement a pilot project to inoculate seeds with rather, there's been an historical strategy to inoculate microbes as, let's say, as probiotics in agriculture. And this is not by no means a new, let's say, technology or idea. It's been, it goes on for over 50 years. But we know that the success rate has not been as expected. And now, thanks to the next generation sequencing, we are now discovering that the communities in the soil and in plant-associated communities are much more complex than we first thought. So really, the project objective here is to implement, let's say, a pilot project inoculating seeds with the entire rhizosphere microbial communities, rather than single strains or consortia of strains. Okay, so really, this is we want to have a platform, and these 8 partners are both institutions, universities, as well as companies, but really to have a platform to investigate and discuss this with different stakeholders like researchers, farmers, agronomists, and biotech companies, in order to see if we can translate these methods and try to develop some innovation in this field. Of course, we are not the only ones doing this. There's now several companies and laboratories around the world that are thinking to do much more like a transplanting approach rather than single strain approach in order to promote agriculture with the use of microbes. Okay. Within this program of CEDRIC, and it is one of the activities of these Interreg programs, is also to divulgate this topic. So we are very happy to organize, and we have to organize, but we're very happy to organize a series of webinars in different topics. And we have 3 topics: soil health and biodiversity, bioinoculants for agriculture, and sustainability and science communication. And today, we are inviting a speaker under the umbrella of soil health and biodiversity. Okay. And these are the series of speakers. We already had 3 speakers, and these are the series of speakers that we have. And you can see here the list, and you can, if you want, take a picture. And today is the 14th of February, and we have. I'm very happy that Davide, who we know each other, and is an very successful Italian scientist who has his own research group in the UK. Davide comes from Italy, and he obtained his PhD in plant biology and crop production from the University of Milan. Okay, working in Milano, but also in other laboratories in Italy. He then went to be as a postdoc. He moved to Germany, to the Max Planck Institute for Plant Breeding and Research in Cologne, which is, let me say, for people who work with plants, is a major world hub for research, where he was extremely successful. Making some very impactful research in plant-microbe interactions, landmark studies. And in 2013, Davide secured a prestigious Royal Society of Edinburgh research fellowship, with which he was able to establish his own group at the University of Dundee. So having said that, I stop sharing and I leave the floor to Davide and thanking him again. And Davide, the floor is yours, and thank you again for joining us.
Okay, thank you very much, Vittorio. Thank you very much for the kind invitation and the kind introduction, and a very good morning to everyone. I'm very pleased to be here, although this is a remote opportunity. I'm very curious to see also the development of your project, which is very, let's say, aligned to what are the research forces of my team. And in today's webinar, what I would like to do is provide you with a brief introduction to the microbiota populating the root-soil interface, prior delving into some of the results that we've gathered using barley as an experimental model. Those are published results, and what I would like to do is giving you a snapshot and emphasizing maybe the rationale or what was behind some of the experiments that we have conducted that may be relevant or useful for your own project. Then I will shift gear, and I will provide again a brief overview of a European project that has just completed, that perhaps is more aligned to the inoculants part and biological parts of SEDIC, but I think it is going to be relevant nonetheless for today's discussion. And then I will close with a couple of slides illustrating where I would like to go in the upcoming future and what kind of impact this may have for basic science and translational application. So now I set my own timer because I don't want to run over time. Okay, so now before doing all that, I would like to share a couple of let's say potential opportunity for networking. One is a conference that is going to take place in Edinburgh this summer, so registration is still open. You have still opportunity to submit abstract. I strongly invite you to do so. And the other one is an action, is a course action that you may be aware of, are networking actions. And again, please visit the website. This may be relevant to your work, and there are opportunities also for exchange, especially for junior scholars. And okay, so my lab is interested in global food security, and global food security passes, at least in part, through soil and the microbes that are present in soil. An area which I'm particularly interested in is the rhizosphere, which is the thin layer of soil surrounding plant roots. From chemical, biological, and physical point of view, this portion of soil is different from the soil that is unplanted. And in this rhizosphere, there is a very distinct microbial community, referred to as the microbiota, capable of interacting with plants. The rhizosphere is also a site for resource exchanges. So plants effectively fuel the microbiota populating the rhizosphere with organic compounds released via rhizodeposition in the rhizosphere. And in turn, microbes there can provide plants with ecological service, for instance, facilitating the uptake of minerals. And from interaction standpoint, the relationship occurring at the root-soil interface are fairly similar to the one that are occurring in our own digestive tract with the microbiota that we are, let's say, the microbiota populates this environment. And what we would like to do in our lab is try to understand a bit more on the interaction between plant and microbes in soil, to be in the position of potentially rewire them for the benefit of the plant and the benefit of mankind. Those are biological processes, and like any biological process, should be seen through the lenses of evolution. And in plants, the hallmark of evolution has been represented by the processes of domestication and breeding selection that have progressively differentiated modern cultivated varieties from wild ancestors. This has changed the genetics and the appearance of plants above and below ground, and this may have impacted also the capacity of engaging with microbes. You may see in wild ancestors of modern cultivated variety and their microbiota perhaps better adapted to marginal soil condition. However, and this represents a potential resource for agriculture. However, breeders, although unintentionally, may have gotten it right and already selected the best combination between plants and microbes for a given soil. And interestingly enough, these two scenarios are not mutually exclusive. So perhaps depending on the soil condition, the more ancestral microbiota may represent a competitive advantage compared to the modern one, and vice versa. But regardless of what is the flavor that you may have or you may prefer of this story, I hope that you'll agree with me that to study this phenomenon, you need plants, a model that has been subjected to the processes of domestication and breeding selection, and at the same time is a genetically tractable organism. And in our lab, we believe that barley is an excellent model to study those processes. It is a cultivated plant, and as such has been subjected to the processes of domestication and breeding selection, but at the same time is an excellent model for experimentation, especially in genetics, for a number of reasons that I listed here. And for when it comes to the microbiota, the fascinating aspects is that the wild ancestor of modern cultivated variety are still present in the areas where barley was domesticated in the first place. There, and some of them are interfertile with modern varieties, so allowing scientists to study biological phenomenon and plant trait within the framework of domestication and breeding selection. This was one of the very first things we've done in our lab. So looking at the composition of the microbiota across evolutionary history, and how we are basically investigating the microbiota is depicted in these slides. So we tend to take reductionistic approaches. So this is where our lab is located, is the James Hutton Institute on the outskirts of Dundee, and this is the favorite way we grow our plants. So under controlled condition in glass house. This allows us to concentrate on the interaction between plants and soil, and leaving out, let's say, other unpredictable environmental parameters. And almost invariably, what we do, we grow plants until a given developmental stage. So we pull them out from the soil, we give it a good shake, and the thin layer of soil that remains at the end of this procedure is what we operationally define as a rhizosphere. Then we have procedure to collect it from the roots and extract genetic material from it. And then this is subjected to high throughput sequencing. And depending on what technology we are using for sequencing, we may address the two questions that I've listed here. And addressing them implies an extensive use of computational biology approaches. And for this reason, I've placed here the link to our GitHub page. So all the figures that I'm going to show that have been published, you can find in that page the scripts to reproduce the figure itself and the stats associated. And also, for the remainder of the presentation, in this portion of the slides, I'll provide you with additional information regarding the stats or the approaches we have followed for that particular. Okay. One of the very first questions that we tried to address in our lab was that whether indeed by growing different different genotypes of barley belonging to different categories, we were able to discriminate their microbiota. And let me guide you through this graphical representation, which is a very common graphical representation when you discuss and present microbiota data. And actually, microbiota scientists tend to abuse of this type of representation, and I am no exception. So you're going to see a lot of those graphs throughout the presentation. So let's see what is in there. So every shape here represents one of the samples that has been subjected to sequencing. And that can be colored or represented according to an independent variable that you have imposed. In this case, whether or not this sample was belonging to one of the three major categories that have been defined by the evolutionary history of barley. Elite variety, modern variety, wild ancestor, and land races that are somehow evolutionary, not in the middle, but basically an intermediate step towards modern variety. Every shape here is placed in a plot according to their microbiota composition, and shapes that are close to each other share more microbes than shapes that are more far apart. And by applying this technique, we were able essentially to predict whether the microbiota per se could inform us on whether we were dealing with an elite variety or a wild ancestor. And this can be supported by appropriate statistical tests. This was a common garden experiment, so everything else was conserved, and the only, let's say, variable was the genotype, suggesting that indeed we were able to reveal such a footprint of human selection on the microbiota. And we went on and looked at multiple accession in the Southern Levant, which is an area where barley has been domesticated in the first place. And what we have revealed is that there is not only a differentiation between elite and wild accessions, but within elite and within wild ancestral, you have different groupings. And some of them may be the result of adaptation at different climatic or soil conditions. So this motivated us to question whether the capacity of barley plants of shaping the microbiota was hardwired in their genome or could be modulated by soil conditions. And we decided to focus on nitrogen and nitrogen availability for plant uptake. As you know, nitrogen is the major rate limiting factor for crop productivity globally. Barley is not capable of engaging in symbiotic association with nitrogen fixing rhizobia, so nitrogen has to be acquired by soil in different ways. And at the same time, mismanagement of nitrogen fertilization may cause, let's say, as negative consequences for the environment. So there is an interest on trying to understand whether this process can be optimized, especially for small grain cereals. And the idea that we had in mind was very simple. We have taken an elite variety and two sessions, wild barley sessions that were contrasting for the capacity of engaging of the microbiota. And this magenta, green, and yellow is going to be the color coding that I'm stick to for this part of it. We have subjected the plants to 3 different nitrogen treatment, keeping all the other minerals at an optimum level. So we are creating, or we are trying to creating a nitrogen deficiency in this way. And 100% means the optimum dose for, let's say, cultivated barley. This is a quarter of it, and 0 means that we did not apply nitrogen to the system. So we have forced the plant somehow to thrive on or try to engage with microbes and recover nitrogen from soil. The first thing we tried to, we wanted to ascertain is whether we have created a nitrogen deficiency system, nitrogen deficiency in the system. And both in soil and in plant, we could see that depending on the treatment, the amount of nitrogen and the performance of the plant was responding to an increased amount, suggesting that under the 0% condition, we have a nitrogen deficiency in our system. So we went on and looked for the microbiota composition. Recall, these plants here, in previous experimentation, demonstrated a diversification of their microbiota in the very same soil. Now, when we were looking at the composition, we stumbled upon an interesting observation. So this is an upset R plot, and is another representation of the microbiota. For the sake of this presentation, the important bit is looking at the bars whose height reflect the number of bacteria differentially, let's say, accumulate or differentially assembled in a set of pairwise comparison between elite variety and wild barleys at different nitrogen level. To cut a long story short, we observe when the nitrogen is limited in the system, we have the maximum amount of diversification between the genotypes. So the plants were rushing to assemble what would be the most adapted microbiota for that given environment. And we hypothesized that this was relevant for the adaptation of such a condition. The other interesting observation that in the very same glasshouse, in the very same soil, the very same genotypes, when provided with the optimum dose of nitrogen for cultivation, their microbiota becomes almost indistinguishable. Okay, so this is something reminiscent of what has been seen also in other type of symbiosis between plant and soil microbes. For instance, if you think about rhizobia and nitrogen fixing rhizobia legume, if you provide mineral nitrogen, this type of interaction is inhibited, and so is the interaction between arbuscular and mycorrhizal fungi and plants when phosphorus is not limiting the system. So it's part of probably a broader evolutionary relationship between plants and microbes. So we went on, we applied something that is called metagenomics to look not only at the individual membership of the microbes, but the function that they may code, in particular at nitrogen 0, where the differences between the elite variety and the wild ancestor were more marked. This type of graphical representation is the same as I illustrated before, but the basis of the diversification here is not, as said, the membership, but the function that the microbes may code for plants. And again, we can separate the elite variety from the wild ancestor and from the unplanted soil. So this is essentially the plant effect on those communities. We went on and tried to identify the individual categories of function that were responsible for this diversification. And basically what you are looking at here is two sides of the same coin. So you can separate what is predominantly accumulated in the unplanted soil and identify what is the rhizosphere effect. So a functional specialization of the microbial communities associated with plants. And within this, you can further separate between, for instance, what is what is enriched in the elite variety compared to the wild ancestor. And one thing that we observed here is that, for instance, microbes that are thriving in association with an elite variety seems to be trained to be more competitive and dial with the plant immune system of the plant, as opposed of microbes that are thriving in association with wild ancestor that seems to have a more diverse more diverse functional specialization. So we wanted to know whether this diversification had an impact on the capacity of the plant in thriving under marginal soil conditions. So what was different. And we had in mind was the experiment that we set up, it was a so-called plant soil feedback experiment. So we have grown plants, the elite variety and the desert accession, because those were the more distinct microbiota that we could observe in our experimentation for one generation. At the end of it, we kept the soil that at that point would have been conditioned by the material that we was grown in, and we take another, let's say, the very same elite variety and grow this variety in this soil. And we contemplated two treatments. So one we call native, basically the soil as was at the end of the first cultivation, and then heat treated. So basically the same soil that was subjected to an autoclaving treatment. The idea was that the heat treated one, this process of sterilization would have not created a sterile environment, but rather would have disrupted so markedly the native microbiota that if this would have been a contribution to plant performance, should have been detected in the plants that we were growing there. So once we have performed this experiment, we use above ground biomass of our plants as a proxy for plant growth. And indeed, what we have observed is that having a native microbiota rather than a disruptive one is better for the plants. So plants were growing better. However, there was not much of a difference in the term of plant performance, whether plants were, the scenario that plants were exposed to an elite microbiota or a wild microbiota. And we wanted to know what was the cause for that. So we went on and we sequenced the microbiota of the second generation. And this is what we have observed. Again, this is another representation of the microbiota. We are looking at here at microbes that were enriched in the elite variety exposed to this different soil. And what we have observed, there is a group of bacteria belonging to different taxa that was significantly enriched in the rhizosphere of barley plants when grown in our soil, when compared to the bulk soil, so basically the unplanted soil, or when compared to the microbiota of an heat-treated soil. So plants were incapable of recruiting those microbes in that particular condition. However, if you look at the composition of this plot, what you see is that regardless of the input soil, the elite variety that we have tested tend to recruit the same microbiota. So we derive two conclusions from this experimentation. First, this may represent the potential probiotic that barley plants tend to recruit from, from soil when grown under nitrogen limiting condition. And the second, and perhaps for us most important one, in this particular scenario, plant genetics was ruling over soil ecology, because regardless of the distinct input communities offered, the elite variety converged on what they essentially like the most. So to try to then resolve or gain insights of the potential of the wild microbiota for elite varieties, we decided to set up a new experiment, whereby we were looking at the genes of the plant regulating the microbiota, because knowing those would have allowed us, in the longer term, to basically reassemble a wild microbiota in an elite plant. And this is the scheme of the experiment that we have performed, and that is essentially an association mapping experiment using the microbiota as an external phenotype. And this slide reminds me also of the fact that I'm going to change the color coding from the reminder of this presentation. Whenever you see blue or shades of blue, I'm referring to elite material, so modern variety or modern microbiota. Whenever you see yellow, I'm referring to the wild material or the wild microbiota. Okay. First thing we started was looking at the segregation of the microbiota within a progeny between a cross between an elite and elite variety and wide section. And this is the type of plot that we've obtained. Now you're fully expert and competent in analyzing this. As you can see here, we have the microbiota of the elite parent, the microbiota of the wild parent. And what you can't see, though, are a categorization of the, let's say, of the microbiota of the progenies. Actually, the majority of them seems to be closer to the elite variety. And when you see this result, we thought that this could have, for us, represented a nice confirmation of our work, because the population we used is designated as BC1S3. And if you are not familiar with this genetic terminology, essentially the geneticists that developed this population did two things. So first, they create an hybrid between the wild ancestor and the elite variety. And this F1 hybrid was crossed again with the elite material prior giving rise to the progenies by self-propagation and therefore the entire population. By doing this procedure, by doing this back cross, the proportion of the elite genome in the population is greater than the one of the wild one. And therefore, if the genome drives the microbiota, then it makes sense that the majority of the microbiota of the population is closer to the elite parent. However, the fact that we didn't see such a categorization allow us to basically consider this phenotype as a sort of external quantitative phenotype. And we apply, therefore, quantitative genetics theory to try to identify whether we can pinpoint position in the barley genome supporting the accumulation and recruitment of certain microbes. And this is what we have done. This figure has two pieces of information in it, so let me guide you through it prior, let's say, analyzing the results. So what you are looking at here, the inner part, these 7 blocks in the inner part, this is the barley genome. Each one of these lines represent the molecular markers. And then you see that we, let's say, superimpose to this 7 chromosome, there are 3 layers representing 3 taxonomic groups of the microbiota. And basically, there are connections between members of these 3 layers and the barley genome. So those connections represent a significant association between one or more groups of the microbiota and a given position of the barley on the barley genome, suggesting that the abundance of these microbes is related to that, to having that particular or having particular alleles at that position in the genome. And with this key interpretation of interpretation in mind, I hope you agree with me that there is a position on chromosome number 3 that we have defined as a locus QRMC3HS, and for the remainder of presentation is going to be called locus 3S for simplicity, which may represent a paradigmatic example on how the barley genome may shape the microbiota, because it's significantly associated to multiple taxa at multiple, let's say, multiple phylogenetic diverse microbes at multiple taxonomic level. So when we observed, when we obtained this result, we decided to challenge it and see whether indeed this locus may be a determinant, of the microbiome. And the experiment that we had in mind stemmed from one of the observations we made in our population, that there were individuals that have residual heterozygosity at the locus of interest, meaning that upon a procedure of selfing, we could have generated lines that were either homozygous for one allele or homozygous for the other allele, while the remainder of the genome was largely conserved. They are not true isogenic lines, but the majority of the variation lies on locus 3H between these two lines. So effectively, we try to Mendelize the QTL that we have identified. The null hypothesis we had in mind was simple. So if locus 3H is let's say a modulator of the microbiota, having different alleles may shape the composition of the microbes. And what we wanted to do is basically the null hypothesis, that we have set up, is that there was no effect whatsoever. So regardless of this allele, the composition of the microbiota would have been identical and similar to the one of the elite. So we were in position to reject this null hypothesis, because when we grew these sibling lines in the very same soil where we have performed our mapping experiment, we could separate microbiota composition, at least for what concern the bacterial component. At the time, we were working with a relatively limited number of plants, so we decided to test also the fungal component. And in that case, we did not see such a separation. So the community were more overlapping among lines, suggesting that locus 3H may operate a selection alone, is sufficient to operate a selection of the microbiota. However, at least in the condition in the soil we tested, predominantly from the bacterial one, not in the fungal one, something that has been observed also for other determinants of the microbiota in other species, for instance. Okay, so the locus contains multiple genes, so we wanted to identify maybe candidates that may operate this selection, and we reasoned that genes that are expressed at the root-soil interface would be ideal candidate for the selection of microbes in the rhizosphere. So what we have done is basically taking the roots that were the leftover of the microbiota survey that I showed you in the slide before and subject them to RNA-seq experiment and try to identify genes that were differentially regulated between the lines that we have tested. We identified a number of genes differentially regulated between these two lines, and the number, maybe if you're familiar with RNA-seq experiments, are remarkably low, 3 orders of magnitude lower of what is considered the number of genes that barley may code. But keep in mind, we are dealing with sibling lines that whose genome is largely conserved. They are actually, let's say, differentiate predominantly for what happens at locus 3H. So it makes sense that their transcriptome is largely conserved and the variation may be limited to the genes that are mapping at the locus of interest. So we decided to set up then a series of criteria to further select among the genes differentially regulated the one that could be potentially implicated in shaping the microbiome. So first and foremost, we wanted genes mapping at the locus of interest. We know where to look for. We are seeking a marked differential expression because marked differential expression may be an index of presence-absence. Recall, we have mapped the genome and we are now trying to identify the candidate using the transcriptome. Genes may be expressed at the very same level, but there is a polymorphism in their sequence leading to different or potentially different proteins. And this is the causative action that ultimately shapes the microbiome. We were able to identify genes belonging to each of these 3 categories. And once we have obtained this list of top candidates on the plant side, shaping the microbiota, we went back and looking at what was happening in the microbiota, so the function operated by microbes to colonize certain environment. And coming from different 2 angles, we found rather striking that we have immune receptors among the candidate genes of the plant when microbes need to, let's say, adapt to what is the plant immune system of the plant. Or we have genes like the XTH, which is operating essentially a modification of cell wall, while microbes are adapting to, let's say, to the metabolize carbohydrate. So we found this particularly interesting, a sort of confirmation coming from a set 2 different angles that we could identify or we could resolve the genetic network linking events on the microbiota, and therefore we decided to move forward, concentrated in 1 candidate, and we wanted to start with the NLR, which represents a plant immune receptor. Before diving further into it, I would like to provide you a snapshot of the plant immune system and certain aspects that are critical for the remainder of the presentation. So plants do have an immune system like human, but unlike humans, they don't have an adaptive component. So each cell is designated to defend itself, and it does so by using essentially 2 main categories of immune receptor. One of those is intracellular and codes for the so-called NLR that are historically been involved in the recognition of effectors secreted by microbes within the cell, and then this will lead to the initiation of immune responses. However, it's important to consider that plant immune responses have a cost, so their activation has a cost, and if this is misregulated, may drag resources for the growth of the plant. So a growth defense trade-off has been previously identified in plants. Okay. With this in mind, we can now look at the NLR, and the reason why we were very keen on starting the characterization of the NLR is that when we were looking at the distribution of this candidate gene, or in general the locus, across multiple barley varieties. So this is the parental line of our population. This is another variety which is very well studied in barley genetics. And if you line up the sequences of the locus, the white one is our locus, the gray one are the boundaries, what we identified is that the locus display a structural variation, and the NLR gene, so our candidate maps in this area of structural variation. So there is a polymorphism. And we wanted to know whether this polymorphism was dictated essentially as a consequence of the evolutionary pressure and selection that has occurred in barley. And so we went back to existing data and tried to characterize, based on molecular markers, whether a particular barley variety had a 3H-NLR-like Barke. Barke is our elite parental line of the population, or like a potential wild ancestor. And then we have, upon this characterization, we have separated between plants and barley varieties that had either 2-row or 6-row. This is important because the separation between 2-row and 6-row dictate a number of traits that are different between barley varieties. And if we had a perfect alignment between 2-row and 6-row and 3H like Barke or 3H like wild, this probably is a consequence of this evolutionary pressure. But what you can see here is that you have yellow and blue dots in both categories, suggesting that there is another element of selection that has operated on 3H-NLR. And very often when it comes to cultivated variety, the element of selection is yield. So we went on and analyzed the existing data of yield for a number of barley varieties growing in a soil that was fairly similar to the one we used for microbiota investigation. And we categorized the variety as, again, as whether they have the Barke allele or the wild allele. And what we have observed is, at least for us, surprising at the beginning, is that plants that had the wild like allele were more productive if categorized according to their haplotype at locus 3H, were more productive than plants that had the Barke allele. And this reminded us of a potential trade-off between growth and defense. So we went on and test the expression of pathogenesis-related PR genes that are bona fide marker for immunoactivation in plant tissue, and we decided to test it in roots. And what we have observed is that the cultivar Barke, our parental line having the immune receptor, as an expression of these genes that is, let's say, higher, significantly higher than the sibling lines carrying the wild-like locus, and also the sibling lines that is, and also the sibling lines carrying the allele of Barke is more, let's say, a significantly higher expression of this gene compared. This suggests that there is a sort of immune activation that is dictated, at least in part, by the locus 3H, although other or additional factor within the plant genome may modulate this phenomenon. And what you can see here in this graph is another color, the color green, which is another variety called Golden Promise. And in a second, you'll see why we decided to introduce this additional element in our comparison. And Golden Promise behaves in a similar way compared to Barke. So there is an activation of immune responses compared to material carrying a wild allele, although not to the same extent. Probably there are other regulators in the gene. So the variety Golden Promise is here. Shares the composition of the locus with the variety Barke, and also the presence of the 3H-NLR. So there is no gap. They are identical. And we went on and decided to investigate what was the microbiota of these two varieties. And in this case, we have yet another representation of the microbiota. And here, in this triangle, every dots represent one members of the microbiota that we have identified in our sequencing survey. What size and the size of the dots, reflect the abundance of that particular microbe in the sequencing survey. And the position reflects where those microbes were identified or preferentially identified. So here we have the implanted soil, here is the rhizosphere of Barke, here is the rhizosphere of Golden Promise. You see a series of dots towards the baseline. So this is the rhizosphere effect of the plant. So those are the microbes enriched in the rhizosphere of those two plants. And as you can see, the majority of the dots is alpha weight. There is no significant difference between the two varieties. So the same locus, the same microbiota. Now, why this is important? because unlike Parker, Golden Promise is a variety that can be efficiently transformed genetically. So what we have decided is taking let's say an attempt at genome editing. So this is a cartoon depicting our 3H-NLR candidate. We have designed a guide RNA at the very beginning of the gene, and we wanted to know whether we can create mutation using these techniques of genome editing. And we were actually quite lucky because we identified lines that carried a significant deletion, 28 base pair in the sequence of the gene. And this deletion at the beginning disrupt the sequence downstream and very likely disrupt the functionality of the gene. And because of this relatively macroscopic, if you want, modification, using a simple electrophoresis gel, it would be possible to categorize plants like this one, harboring the mutation, so the fragment is small, from the what is wild type situation, the fragment is larger, and also heterozygous elements, in the sense that this may be the result of, let's say, heterozygous members of the transforming population. So the system worked, we managed to obtain edited lines, and this allows us to test this hypothesis that I depicted. So we hypothesized that Golden Promise has its own microbiota. So we have control that was subjected to transformation, but did not have this deletion, so is a failed edited attempt. But this is important because the transformation is also another parameter that has modified those plants. And this one we hypothesized that would have carried a microbiota more similar to the wild type plant, as opposed to plants that were subjected to the transformation and the editing that would have a different microbiota if 3H-NLR is ultimately the driver of this modification. And then we went on and we grew these plants. And I'm very glad to present these data here because these data are the result of University of Bolzano very own Sonia Monterisi, who at the time was a PhD in my lab. So plants did not have a pleiotropic effect. We could grow them in soil, we could grow them at homozygous state. So we know, we have a marker, we know which one were carrying the mutation, which one were not carrying such a mutation. And ultimately we went on and looked at microbiota composition. Here, those bars reflect the major groups that we have identified in the microbiota of those plants, again, partitioned according to the genotype that we have tested. And what you can see here is that, indeed, if we look at the transformed and edited plants, we have a decay of members of the actinobacteria that is associated with an increase of member of proteobacteria. This shift between actinobacteria and other members of the community is one of the hallmarks that differentiate the elite variety microbiota from the more ancestral one. However, the devil is in the details, and as you can see here, our controls do not have a phenotype that is overlapping Golden Promise, somehow intermediate between the two, suggesting that perhaps transformation per se has an impact on microbiota composition. And basically, this is what we are investing or try to addressing at this in this moment, try to creating a genetic material that allow us to separate the two events and then having a better understanding or firm grasp on the impact of 3H-NLR on microbiota composition. So watch this space. No, sorry, this is the previous one. Okay, so now let me shift gears. So last 10 minutes of this intervention, I would like to introduce, provide you an overview of a project, European project that has just completed and was looking at the microbiomes across plant food chain as a way of predicting and better using these food chains. I had the pleasure to be a working package leader in this project. And I was leading the working package dedicated to the plant food chain. So we had the opportunity to investigate the microbiota of several crops. Essentially, we had the three questions that we were asking in this project were the similar questions that I've illustrated before. So composition, function, and whether we can shift the composition and function in order to ameliorate the productivity. As I said, it was a very successful project. So we have quite a number of papers out. Not all of them are full merit of my own team. So colleagues of Luke in Finland and especially University of Bologna when it comes to grape wine made a significant contribution to that. And today I'm going to talk about spinach because, as you can see here, there is no paper yet, so we are still developing this story, and we think it's a quite interesting one, and I would like to share with you. So this, unlike all our barley research, this investigation was actually an investigation conducted in the field, in actual farm. So we engage with farmers and we sample microbiota, and let's say we sample plants and microbes in their farm. So we wrap up everything and ship to the United Kingdom, where we then process it for microbiota composition. There was a number of samples here. I mean, this is just a snapshot. This is a relatively large microbiota investigation. So we thought it was important having a sort of throughputness in terms of processing this material. So we trust more people than machines. So this is our high throughput processing system, to a phenomenal technical assistant that helped us throughout those years in preparing thousands of samples for microbiota processing. And this is probably the only very technical slides that I'm going to show you, because we were dealing with samples harvested at different seasons in different farms. We rely on technical replicates within library and between libraries to try to identify control for reproducibility, because otherwise the library effect would have confounded any effect of the biology that we were interested in. So we're going to skip rather quick about, let's say, rather quick on this part, but it's important to keep in mind that if you want to work with samples that are sampled on time, you need to have this control to infer meaningful, biological meaningful conclusion. So with this in mind, we started to look at the composition of the microbiota, the bacterial microbiota of spinach, across actually 4 microhabitats. We went on and looking at the leaves as well, because this is the main, let's say, the main output when it comes to sampling and processing of the plant. And now, if you can see and look at the shapes here, what you can appreciate will be a microhabitat effect from the unplanted soil to the roots and leaves that basically differentiate the community associated with spinach, suggesting that there is a plant effect in shaping this community. And the gradient is conserved between the farms that we sample in the north and in the south. So there is something that is hardwired in spinach plant, regardless of the environment and the other factors that may influence those farms. So we decided that we wanted to investigate and try to identify in existing collection bacteria that were related to these regulators of the spinach microbiota. We sequenced their genomes, so those were said phylogenetically related, were not exactly the one that we have isolated from the farm, but they were basically the second best option, and used those to try to manipulate the microbiota using a more conventional approach, as alluded by Victoria in introduction. This is something new. So the application of microbes in agriculture has a long history. What we wanted to do is essentially test 2 types of intervention. One at the beginning, at sowing, because the plants would have developed together with the inoculum, and another one that was applied later on, a sort of booster. The spinach cycle is relatively short. It can be as short as 2 months. And then we went on a week before harvest to sample the plants, and then the plants were harvested, and when we measured the let's say the biomass. So the idea is that if those are the regulator of the microbiota, if we supercharge them at the beginning of the cultivation, we have a more stable and more rapidly established endogenous microbiota that may better withstand environmental fluctuation. So this is the theory, this is the practice. So we reasoned that we need to apply those inoculants in using an approach that was already established in the farm to reduce the cost and ensure that farmers may apply it. Just doesn't work. Doesn't matter. There will be a tractor going and spraying a product on the field alongside our bacteria. This works. Okay, I have to cut short because otherwise I'll be running out of time. But essentially, this is what happens. I just move forward, and we went on to see whether there was any impact on the productivity of the plant, because ultimately this is what matters. Probably what matters the more for farmers. And the 2 treatments now designed by these 2 colors were not significantly different between them. However, what I'm going to show you here, is the yield difference. So the yield gain compared to the control. The control were basically the same strip of field subjected to the same treatments, sorry, to the same agricultural management as the treated one, without, of course, the application of microbes. And if you do now this calculation, you can appreciate that this has resulted in a positive impact for the farmers, because I could finally, after many years that I was saying that our type of science, our science would have benefited agriculture, we can effectively give a price tag. Now, will it work in any spinach farm? Will it work in any spinach farm in any condition? I can't answer this question. In the one we tested in the north and the south of Italy has provided us with a positive result, and we wanted to know what was the cause of this positive result. We went on and look at the composition of the microbiota in the leaves. And again, the idea here is looking at whether we identify first our inoculants, and to our surprise, we were not able to identify an increase of the population of microbes of the one that we had applied. But what we have observed in the leaves of those plants is the decay of certain categories, such as Erwiniaceae or Pseudomonaceae, that are often associated with a sort of dysbiosis of the microbiota. They may cause yield losses even in spinach. So it's like the application of our inoculum. One of the hypotheses that we are exploring is that as either activated responses in the plant or response in the microbiota to keep at bay potential pathogen, and consequently this has led to an increased biomass. However, we wanted to see this picture from a broader angle, so the productivity is just one thing. There may be another impact of this application in the environment, and we were particularly mindful that playing with microbes may lead to let's say a potential fluctuation of IMR genes in the environment. So we decided to look at the composition of this IMR once the spinach arrived at the processing plants, and in this case using the wastewater as a proxy for IMR dynamics. What you can see in this heat map here are a series of IMR genes, and this is their, let's say, the color coding reflect their abundance in our samples, and the samples are categorized according to whether either treated or controlled, or whether they were belonging to the first washing system or second one. And to cut a very long story short, you don't see a separation according to color, so there is no difference between what would have been an ordinary spinach field and the treated one. The only difference is according to shape, and makes a lot of sense. So depending on the washing, you may have a different residual of what would be the endogenous IMR titer. So basically, we have improved the productivity without really messing too much with other environmental parameters. Again, in the farm and the condition that we've tested. And this is basically what we are now elaborate, or we try to wrap up in terms of story. So watch this space. Okay, this is the end of my intervention. So what I show you today is that the microbiota may act or may be treated as an external plant phenotype. There are ways that microbiota can be manipulated. This is either via genetics or via supplementation, by application of inoculants in the and there may be an impact on productivity. And as said, and I would say limited to what we have seen, there are a number of variables that we haven't tested, and we can comment as to whether it may influence the dynamics. So where we would like to go, there are a number of questions that we are still want to address. So an interesting aspect of this potential probiotic of the plants is that they are already installed, and very often they are already associated to plants. So why plants are getting sick, or why plants are not acquiring minerals at all time? So maybe there are trade-offs that from molecular, let's say from molecular standpoint, have to be clarified. It would be interesting, or this is something that we would like to explore, is whether using microbiota as a predicting tool for plant breeding. This may be particularly relevant for species, so forgotten crops or orphan crops, for which there are no genomic or genetic resources at the moment when it comes to adaptation to the environment. And if you want to really do so in a transformative way, probably we need to look also at the way we are testing or characterizing our microbiota, maybe using techniques that can be applied in the field. For instance, Oxford nanopore could be, again, a game changer towards a more swift application of this. Where we want to go, well, I'm nearly there, is basically something that I'm rambling about since the now a decade, and basically using and closing the circle with the parallelism between medical science and agricultural science. So using microbiota information like a tool towards a personalized agriculture, and therefore try to address a series of potential targets, not only the productivity of the plant, but more general the carbonization of the agricultural system. Okay, so this is the end. I would like to pay my tributes to my co-workers and the funding bodies that have supported our work. And the very last one is for you. Thank you very much for your attention. I'll be happy to take any questions.
Thank you very much, Davide. I was really glued. Sometimes, you know, when you listen to webinars, you kind of open your email or you are wandering with your head. But you really glued me to your presentation. And, you know, being a microbiologist is so, I must admit, it's rare to hear talks that really bring in the plant so much into the equation. And I think we need to do much more work together, microbiologists and plant scientists. And I think you're doing both, and it's really, really very interesting. So I want to open the floor for questions, and I already have a few in the chat. I also have a couple, but let's start in the chat, and I would like to try and make them according to your presentation, not to mix the sections too much. So Salma, hello Salma from Morocco. She's a PhD student at the UM6P in Benguerir. She's asking you whether, you know, she's going to the 3H story, and she's asking whether you, apart from your RNA-seq, you also did some molecular metabolomic GWAS in that root bacteria interface.
Yes, we did. And we did not identify any particular signature. I think I have a slide here. Yeah, so we look at the multiple element of root system architecture, and there was no real difference between the sibling lines. We went on and look at the exudates. We look at predominantly at primary metabolites. We haven't looked at secondary metabolites, so we can't really exclude that is an effect of that. But looking at the primary one, we couldn't find a significant effect.
Good. So, I mean, I have a question which I would like to ask now because it's linked to Salma's question. And did you do like RNA-seq, bacterial RNA-seq, in order to try?
No.
No.
We
Okay.
haven't reached for poly-A. We just went for plant transcripts.
And RNA-seq, I mean, you focused on that area. RNA-seq in the other part, in the totality of plants. Did you?
No, we haven't. We haven't done that. We have just focused on the roots.
Yeah, I appreciate to be focused. Otherwise, with all this, it would. Okay.
No, it's a fair point. I mean, this is part of the very long to-do list.
Yeah, of course. Yeah. Also sticking to the same 3H story, Tariq Shaha is asking whether you see any pleiotropic effects of the 3H on the plant.
Yeah, it's a kind of tricky story because so we went on. The first thing we have looked at was the yield of the plant, and both the sibling lines did not differ between. So the sibling lines had different pH alleles, but they don't differ in terms of yield, and they are still different though from the elite variety. Now we realized that this could be due to the fact that this is material derived from wild ancestor, and both sibling lines have this gene. It's called BTR at the wild status, which is the brittle rachis. So meaning that this is probably their yield can be properly assessed due to the presence of this gene that is not very far away from locus 3H. So what we have done now is try to prune out this locus, and the same holds true for the transformed line. So we want to have more clean material and see whether this has an impact on yield. So it's not yet conclusive. I can't really have an answer on that other than the other varieties that I show you.
Okay, thanks. And Salma is another question. And Salma, since you have 3 questions, why don't you connect and show your face and ask, so you can ask it yourself. Salma.
Yeah, I can do it. Sure. I'm here.
Oh, hello, Salma.
Yes, hello. Thank you so much for this insightful presentation and for your achievement so far.
Thank you.
Well, I have many questions. Well, first, my question was related to actually MGWAS, and I was like targeting not only the metabolomic parts, metabolomic signature, but actually even like the microbial part. And like since you were doing like a lot of things and you were adopting a holistic approach, what you did not do like a multivariate analysis, like just to not to analyze separately each approach independently, but actually to do it in a holistic approach. Actually, I did like a presentation recently in the journal club related to this like approach, that they were like combining all actually the approaches, and they were like achieving a lot of like output so far, and a good result so far too. So I don't know if you can like.
Yeah. No. I mean, I will briefly comment. Oh, sorry. Go ahead.
I don't know if you got the points, but actually the purpose is just to do the multivariate analysis instead of doing it like separately.
Oh, sounds like a bit of artificial intelligence. Does it sound like a artificial intelligence project, Davide? What's she saying?
Yeah, no, I see. I think that the rate limiting step probably is the funding of getting number of plants profiled for, I don't know, metabolites, microbes, genes. I mean, it's ideally is fantastic. So we took a more pragmatic one because the funding was, I'm not saying limited, but did not allow us to take this approach. We reasoned that if we start with sieving this population and try to identify lines that are representative in terms of microbiota characterization, those should be the one that we should focus more, because then you reduce the number of plants to something, let's say from hundreds, if not thousands, to a handful, and those can be then characterized in a much greater way. Of course, there is the limitation here is that you are taking the assumption that your lines, like in our case, these locus 3H, are the, let's say, the most significant one or the most important one, and maybe there are other traits in the genome that we have, let's say, we will leave behind in this way. So we are aware of that, and if funds becomes available, if funding becomes more funding becomes available, then certainly this is a very interesting approach.
Okay, that's perfect.
Thank you, Salman. Nice seeing you. See you on Monday.
See you too. Thank you so much.
Bye. Okay, we have a coffee on Monday. Good. Any other questions from the? We have one in the chat. We now shift Davide to the last part of your talk, and Tariq asks whether the dysbiosis that you've seen in the phyllosphere of spinach facilitates the spread of AMR genes among the microbial population.
Yeah, we haven't tested this properly, so it's basically this is a snapshot of what we have collected. We can't really profile with metagenomics the actual leaves, because the amount of plant DNA is as such that becomes, how should I say, it becomes not feasible. So you end up with a lot of plant DNA and very little microbiota. So that's why we profiled the let's say the wastewater, the washing of the leaves. We have tested though also the AMR in the roots, because this is something that we can, in the rhizosphere, something that we can profile. And again, there is no real sign of potential spreading or a differential organization of AMR genes if you look control with treated samples. So the I should say the endogenous microbiota and the environment still rules the organization of AMR and the resistome, and the application hasn't perturbed it yet. The application of inoculants hasn't perturbed it.
Okay. It's also another question regarding still on the last part, just a technical question on the CFU that you apply and your application of the consortia.
Yes. Oh, I don't remember by heart, but I can certainly look up this data and provide you in a separate email. I should have it somewhere. Yeah, I need to look up in our data. I can't reply this question straight away.
So, and also, Davide, you've put like a value 0.22 euro per. What was the
yes.
That's very interesting. That takes into consideration the cost of the product as well, or is it just a purely?
No. This takes into consideration the cost of the product as well. So this 500 would be the gain. There is the sense that the application, so the major cost, so the fuel, the machinery, the application of other products, is already included in there, because the fields are basically strips, one after the other. The farmers are just going. The application of the product is a fraction of it. So we are calculating, there is a group in Finland that is calculating the life life cycle assessment, also other parameters. But yeah, this is give or take a ballpark of what will be the gain. The actual number will be released soon.
And also as a follow-up question, which fits the Cedric project, because we have farmer associations, we have different stakeholders. Can you comment on the acceptability by the farmers or by farmers association of a potential microbial applications? Do you have any comments on that? Do you think?
Yeah, I mean, our experience was extremely positive. So
Right.
farmers were very involved, very interested in this type of approach, because I mean, we were intervening at a time where we had there was the drought of 2022, the increase of cost on fertilizer and fuel due to the international, let's say, due to international crisis. So it was a time where the margins were particularly limited. And certainly there is an interest. Certainly there is an interest in understanding what we were proposing. So it was not really a top-down. We sat down together and see what would have been the best way. For instance, the application of sowing was their idea, because they thought that it would have been more applicable, and probably from our standpoint, a direct exposure to the young seedlings, to the microbiota. So there was a participation. And of course, and especially when working with, let's say, working with real farms, you need to keep in mind that this is their livelihood. So we are doing experimenting. They need to get a return out of it. So we had a sort of insurance whereby if the treatment would haven't worked, or when the treatment maybe hasn't worked in the way we anticipated, there was a sort of payback. So that the gap would have been refunded. So this is something to keep in mind.
Yeah, that's a very good approach. And I know there are some companies connected as well. I've seen Wolfgang, my colleague, that has a company in Germany. So I like this approach as well. So basically promoting and encouraging the use of these products.
Yeah, no, I learn a lot.
I think approaches like this, where we can, not just simply say buy it and that's it. No, but approaches whereby there's a follow-up. There's a follow-up of the use and continuous interaction with the farmer from companies that produce, I think, is very important. Any other questions or any comments? Well, if not, really, Davide, I really thank for your time, and I hope we can connect soon. And I'm sure you created a lot of interest and networking among we had almost 60 participants from all over, and also I've seen the names from all different stakeholders, different scientists, different countries. So finally, before I leave, I just want to remind all of you that we will have a webinar already next Monday. Sorry, it's going to be a short, only a few more days, but we rely a lot on the availability of the speakers. And it will be from Professoressa Moscatelli, and it will talk about communication in soil. So again, still sticking on the same subject. We also have speaker Davide. We also have Zalem from Canada. Obviously, he's in the middle of the night. He thanks, and it was worth to get up, he says.
Yeah, it's very kind. It's very kind. I'm glad that they stayed up until now for this.
Yeah, okay. Also, Silvia from Orogel. Hello, Silvia was connected as well. We collaborate with her as well. Okay, so I thank again, Davide. It was a pleasure to see you and to listen to your really nice, going from bacteria to plant to genome editing. Congratulations for, putting together all these approaches to try to understand plant microbe interactions. So thank you again, and hope to see you soon. Okay, bye-bye.
Very welcome, and take care. Have a nice weekend. Bye-bye.
Bye, everybody. Bye-bye.
Thank you. Bye.